Hello friends, good evening. Welcome to Sa Biology class. Friends, today we shall discuss about chromosomes. Before that, I request all of my friends, those who have not subscribed my YouTube channel, please subscribe. Also watch, share and like my videos. The point is that if you want to know about any important topic related to biological science, then please inform me through the comment box so that I can prepare and post that video in my channel. An organism's DNA with its arrays of genes is organized into structures called as chromosomes, which serve as vehicles for transmitting genetic information. Strasburger in 1875 first observed thread-like structures during cell division. Wilder introduced the term chromosome, which is derived from two Greek words. Chromo means color and soma means body. A chromosome is a rod-shaped, self-replicating and organized mass of DNA present within the nucleus of eukaryotic cells. Normally, the chromosomes are visible only during mitosis and meiosis. When cells are not dividing, the genetic material that make up the chromosome unfolds and uncoils into a diffuse network called as chromatin. Chromatins are thread-like. In eukaryotes, they are composed of DNA, protein and a small amount of RNA. There are two types of chromatin, euchromatin which is genetically active and heterochromatin which is inactive. Chromosomes are most easily visualized during mitosis. Each chromosome has a constricted region called as centromere. The location of centromere determines the appearance of the chromosome. Number of chromosomes. The number of chromosomes in the somatic cells of higher plants and animals is termed as somatic or diploid or zygotic number, while in gametes the number of chromosomes is haploid or gametic or reduced. The diploid number is represented by the symbol 2n, whereas that of haploid number as n. The number of chromosomes is constant in all somatic cells of all individuals of a species. The chromosome number is used for species identification and also to trace relationship within a species. The diploid chromosome number varies from 2 in Myrmecia pyrosula and 4 in Haplopappus gracilis to about 1200 in some pteridophytes. In Aulacantha, a radiolarian, the diploid chromosome number is about 1600. In polyploid individuals, it is necessary to mention the ancestral primitive number represented by the symbol X, which is known as base number. For example, in common wheat, Triticum aestivum, the diploid chromosome number is equal to 42, so N is equal to 21. But X is 7. This indicates that wheat is an hexaploid. Shape of chromosomes. As already mentioned, a chromosome is a rod-shaped, self-replicating, organized mass of DNA. Each chromosome has a narrow or constricted region called a centromere. It is the primary constriction which divides the chromosome into two arms. By convention, the shorter arm is known as P arm and the longer one is Q arm. Shape of chromosome is usually observed at anaphase. The position of the centromere varies. It can be median, subterminal or terminal. If the centromere is median, then both the arms of the chromosome become approximately equal in length. Such a type of chromosome is called as metacentric chromosome, which is V-shaped. In many cases, the centromere is subterminal. So here one arm is shorter and the other arm is longer. Such a type of chromosome is known as submetacentric, which is L-shaped. In very few cases, the centromere is present at the tip of the chromosome. Such a type of chromosome is called telocentric, which is rod-shaped. In several cases, one arm of a chromosome is sufficiently shorter than the other arm. Such type of chromosomes are called as acrocentric, which are J-shaped. Chromosome cycle. During interphase, chromosomes are not visible because they are opened into an interwoven mass of very fine thread called chromatin. But as cells enter the M phase, these chromatin threads undergo condensation. As a result, distinct thread-like structures are seen during prophase. After further condensation, chromosomes appear as compact rod-like structure in metaphase and anaphase. But again during telophase, due to uncoiling, thread-like structures are again seen. The cyclic change in the shape and size of chromosome during cell cycle is known as chromosome cycle. Based on the number of centromeres present, chromosome can be classified into different types such as monocentric, dicentric, polycentric, eccentric and diffused or non-located. First one is monocentric chromosomes which have a single centromere. Such type of chromosomes are normal. Second type of chromosome is dicentric which has two centromeres. Such type of chromosomes are abnormal ones. They are formed by the fusion of two chromosomal segments, each of which has a centromere. In this process, the chromosomal segments without centromeres are lost, and polycentric chromosomes have multiple centromeres. And in eccentric form, the chromosome is without a centromere. Such type of chromosomal segments do not survive long. In several cases, the centromere is non-distinct. Such type of centromeres are called diffuse or non-located. This type of centromere is diffused throughout the chromosome, and in such cases, the spindle fiber remain attached to chromosome at several points. Based on the position of centromere, chromosomes can be divided into different groups such as metacentric, submetacentric, acrocentric and telocentric. In metacentric chromosome, centromere is median, as a result of which both the arms have approximately equal length. Such type of chromosome are V-shaped. Second type is submetacentric, in which one arm is shorter than the other arm. As a result, submetacentric chromosomes are L-shaped. But in acrocentric chromosome, the centromere is closer to one end. As a result, such type of chromosomes are J-shaped. But if the centromere is present at the tip, such type of chromosomes are called as telocentric, which are rod-shaped. Different regions recognized in a chromosome. As already mentioned, each chromosome contains a constricted region called a centromere. It is the primary constriction which divides a chromosome into two arms. The longer arm is designated as Q arm and the shorter arm is symbolized as P arm. Before cell division, a protein complex assembled on centromere on each side of the chromosome. This protein complex is called as kinetochore, which remain attached to spindle fiber during cell division. Functions of centromere. A centromere performs two main functions. Number one, it serves as the point of attachment for spindle fibers during cell division, and number two, it is the nucleation center for polymerization of tubulins. And tubulins are the components of microtubules, so centromeres are involved in the formation of chromosomal spindle fibers during cell division. This figure shows the structure of a single chromosome. But during metaphase, each chromosome consists of two symmetric structures which remain attached at the centromere. Each symmetric structure is one half of the duplicated chromosome and is known as chromatids. So the duplicated chromosome consists of two sister chromatids which remain attached at the centromere. During cell division, these two chromatids are separated and become individual chromosomes. Each chromosome is enclosed by an envelope called pellicle. Inside, a matrix consisting of ground substances is seen, and two spirally coiled threads called chromonemata are embedded in the matrix. These two chromonemata are also coiled around each other. As a result, they look like a single thread. At regular intervals along the chromonemata, small dense structures are seen which are particularly prominent during the liptotin and zygotin substage of meiotic prophase 1. The structures are called as chromomeres, which are considered as genes. In addition to primary constriction, many chromosomes have secondary constriction, which is also known as nucleolar organizer region. This region remains associated with the nucleolus during interphase. Secondary constriction contain the genes for 18S and 28S RNA. This region is thought to be involved in the formation of nucleolus. In addition, many chromosomes also have tertiary constriction whose functions are not known, but they are helpful to distinguish between chromosomes. And the part of a chromosome which is present beyond secondary constriction is called satellite. Such type of chromosomes are called as satellite chromosomes. Next, telomeres. Telomeres are the tip of chromosomes which provide stability and also maintain individuality by preventing the fusion between the tip of homologous and non-homologous chromosome. So friends, this is all about chromosome. For more updates, please subscribe my YouTube channel, Sam Biology Class.